Okay, welcome everybody. My name is Andrew Holmes and I'm chair today for this, which is the first of our integrated nutrition seminar series talks for the year. We've got an excellent speaker lined up for today. We're very lucky to have Professor Tim Spector from King's College London. And Tim is probably a name that's known to the vast majority of you here, I would expect, but in my brief foray into Tim's biography, he's described as a genetic epidemiologist, medical doctor, science writer, and now working on the relationship between nutrition, gut microbiome, and health. In a quick chat with him before, he said he has actually switched careers or fields several times, and the highlight was, what did you describe it? Fruit packing or something like that. Washing up in Austria. I can't believe he did that, didn't make the career highlights, but you know. So Tim's, as you would probably be aware, been very influential in both science and in science outreach, and has a number of books on diet and health. In terms of what my understanding of his career has been, it really kicked off in terms of impact and profile with the setting up of the Twins UK study, which I would imagine several of us in the room here have been involved with, and that's been a tremendously influential program and resource for people worldwide. And it was essentially geared to look at interactions between genetic and environmental factors in disease, which is right at the heart of the things that we do here at the CPC, and it's led on into a lot of research on nutrition. Then there was a paper in 2014, which was the first time that I confess that I saw his name, which was in collaboration with Ruth Lay, presumably Jeff Gordon, and colleagues as well, looking at twins as a means of looking at the way the microbiome develops in humans. And that paper started a whole string of really high-profile ones on microbiome and connection with diet and genetics that Tim's been involved in. And I confess also that I was sufficiently intrigued as to the impact of all of that in microbiology that I wondered if Bacteroides theta iota micron, one of the most famous microbes in the gut sphere, was actually named after Tim, because it Greek translates like that. I suspect that's not true, because the name's a lot older than Tim. But I also wondered if the Greek thing came on with Zoe. Was the life the inspiration for the name there with the company that, Zoe is the area where a lot of you would be familiar here. And this is a nutrition and health advice company that's been set up. It's really pioneered a number of things in terms of use of apps for translational applications, but also in terms of an integrative approach to getting omics technologies and microbiome into the way that we try and develop precision medicine, etc. So there's dozens of papers. I think the H index is now well over 200, and the number of citations per year, 20,000 or so. And I'm very much looking forward to hearing about how all of that is translated into the microbiome revolution. So over to you, Tim, and thank you very much.
Well, thank you very much for inviting me, and thank you for those also listening online. And yeah, it has been a journey, and what I wanted to do here is many things I could talk about. It's a huge topic, so I'm going to focus on one small area of it, the microbiome part of it, and really what we've managed to achieve by me helping co-fund Zoe in order to get this done on a much bigger scale than would have been possible otherwise. So in a way, a lot of the stuff that led up to it, the twins, was a precursor to what we could do, but at 10, 100 times the scale, by using a commercial company to effectively get people to pay to participate in research, a sort of citizen science model, because we really did need big numbers. And people thought, I thought I had huge numbers of twins, wow, 15,000 twins, but that's sort of nothing compared to what we need really to grapple with the mysteries of the gut. So I'll dive in, but before I do that, so a lot of the work here initially involved my colleagues at King's, but more latterly a group at Trento, the Sagata lab, which are really the cutting edge lab in the world for microbiome metagenomes. So I'm stealing a lot of their thunder, but they do a lot of the hard work here, in case I don't mention it, which I often forget to do. Right, let's get stuck in. Oops, wrong one. As you heard, I started life after my sabbatical in Sydney in 1993, where I spent some time at the Garvan. Some of you might remember one of the first genes that was discovered, big time, made front page of Nature, was the vitamin D receptor gene. And the Garvan responsible, and everyone thought this is going to be easy. Genes are going to explain everything. Turned out that research was complete rubbish. A lot of the stuff I did for that first 10 years and many colleagues ended up being rubbish because we had very small numbers. And it turns out that genes haven't really delivered everything that we wanted them to do, apart from for very rare diseases. So for chronic disease, they're sort of useful for mechanisms, but it's very different to the optimism we had 20 odd years ago. And I've been studying twins, and I was particularly interested in why twins might be different for certain diseases. That was a big drive for me. But firstly, it was why they're so similar. Can we find new genes? Then it was, let's look at identical twins who are clones and see what's different about them. And really, I was finding absolutely nothing that was different until we eventually, in about 2011, started working to do this twin study, which eventually got published after a few years in Cell with this group from Cornell. And that really changed my mind about what to work on next. So really that was a pivotal moment around 2011 when I first got those results, showing that identical twins had very different gut microbes. And that was kind of weird. You think about it, these are genetic clones. They lived all their life together. They have the same mother, they went to the same school, ate the same food. And the only thing we could find different about them was their gut microbes, because I'd been looking for 10 years, every biomarker possible, and they were all correlated until we got to the gut microbiome. So that must mean something. It must be something about why one gets cancer, one doesn't. One gets an autoimmune disease, one doesn't. Why one has depression, one doesn't. And so that, to me, was a bit of a watershed moment. And I also had a health scare of my own the same year, a little mini stroke while I was climbing at altitude. And those two things came together, and that said, right, I'm going to do not just dabble in the microbiome, I'm going to really do this full time, and then got into nutrition. So that was how it all kicked off from my previous life in genetics and epidemiology. And on this route, just like for the genetics journey, which we thought would be easy and just a few candidate genes would explain everything, it turns out that our microbes and our food are very complicated. I was taught at medical school that food was all about macronutrients. All you need to know was how much percentage fat people were having, how much protein, how much carbs, and critically, what's the calorie intake for the day? That's all that mattered. That's why nutrition was such a boring subject and avoided by everybody. And no doctor ever wanted to do nutrition, and it was basically not taught because it really was very dull, very dogmatic, and only designed initially to avoid starvation after the Second World War. It wasn't really about excess or optimization. And what we've discovered with tools like metabolomics is that food actually has somewhere between 50 and 100,000 different chemicals in it. So rather than dividing it just by 4, you need to think of it as this massive shop full of chemicals that are potentially fuel for our gut microbes, which are probably between 40 and 100 trillion of them. Each of them have lots of genes. We have 200 times more genes than in humans. There's the same number of microbes as probably human cells. But the key thing is that they are like chemical factories. They are pumping out all kinds, thousands of chemicals all the time, not only vitamins, but also neurochemicals and particularly important immune chemicals that are interacting with our immune system as well as our nervous system. So we think the key role of the microbiome is to produce these chemicals that calm our immune system down, talk to it, tell us exactly what the environment is happening, and then signals go from there to the nerve cells on the gut, and they go to the brain as well to tell the brain what to do rather than the other way around. So most of the traffic between the gut and the brain is from the gut to the brain, not the reverse. So it's really changing our whole perception of who is in charge of the body. The Cartesian idea was that the brain is totally separate and controls everything, just like robots to whatever the brain is doing. The truth is really the opposite, that the brain is just another organ. It responds to what's happening in the gut rather blindly, and it often gets it wrong. The other thing is that obviously our body is not only just the Krebs cycle, but it's thousands of other metabolites that are incredibly complicated, and they all need chemistry to drive them. So this system that started with this boring idea of just a few macronutrients and calories, and that's all a nutritionist needs to know, has become this vast system biology problem that is really fascinating when you start thinking of everything as chemicals. And that's really why this whole area, at the moment, we're still just scraping the surface of what we understand about the interaction between the food we eat, the microbes in our gut, and the chemical reactions they then produce in our body and in every organ. So I was giving a talk like this, but to a lay audience about a book I wrote, because when I change subjects, what I like to do is to write a book on it. I go away for a sabbatical, usually to a very nice place like Spain or Barcelona or Sydney or somewhere like that, and just hide for 3 or 4 months, get a draft of that book, just like writing a new grant. And I did this for epigenetics when I was getting into epigenetics, but I did this for diet and nutrition after my health scare, after I'd had this revelation about the twins. And that helped me really focus my ideas. That book was called The Diet Myth, which at the time didn't do very well, but its sales have continued, luckily, as in a way people have caught up with some of those, what they thought were wacky ideas. And it turns out most of them were actually correct. And I was giving this talk, 2 people were in the audience, they come up after it and said, we think this whole idea of the gut microbiome and the potential for personalization is really good. And, let's form a company. And I said, I'm not really interested in just another marketing exercise, because most nutrition companies, as you might have noticed, they spend all their money on adverts, and their website has a few token doctors in white coats and white teeth with stethoscopes selling vitamins, and that's all they do. It's pseudoscience. So I said, you've got to come up with at least $3 million, $4 million, so we can do a baseline study in order to actually get the algorithms to personalize nutrition based on gut microbiomes and a few blood tests. And amazingly, they came up with that money in 3 weeks, and I couldn't then say no. But although I was reluctant at the time, it was probably the best decision I've made in my career, because Zoe was born, this company, which is very much science-based. We spent the first 3 years really just doing research, which is pretty unknown in a startup. We had some great investors, and we knew we needed the big numbers, because luckily, having gone through this with genetics, of how we totally underestimated the scale of what we need, it was even more true in the microbiome, because all of you guys are different. You all got unique gut microbes that no one else in this room has. You probably only share on average 20% of your species with each other. That's quite a big range to then explain all these health issues on without massive numbers, so you can actually sort out the noise. And this has been one of the big problems of the field, is this natural range and diversity of microbes that may have overlapping functions, but are very different when you're isolating. So to cut a long story short, this was the #1 paper that we got in Nature Medicine. It was looking at 1,000 people, mostly twins, giving them identical meals and seeing what happened to them. No one had ever done it before in more than about 60 people. And the results, it was an extremely detailed study. We had the first 24 hours were done in hospital, and then it was done at home. It was a huge exercise, as you could probably imagine, doing a really detailed clinical trial on 1,000 people. And the #1 result that blew me away was this bit here on the right, the fact that when we gave people an identical muffin, which was fatty and sugary, we had a 10-fold difference in their response of blood fats and blood sugar. Normal people. These are not people with diabetes, no major illnesses. So, typical of this room, you will respond very differently to the identical meal. And the fact that that was repeatable and reliable was a really big factor, because up until then, nutrition had said that's not true. They said, one diet fits all. This is the standard Australian perfect diet. You'll all be fine on it. Go away and get thin. Didn't work, did it? So that was the breakthrough. We knew from that point we had the ability to have an algorithm that we could give people based on all the data we were collecting. So anyone, based on their carbs and their fat responses, we could give an idea of how they're likely to respond. And so personalizing diet is in the very first iteration. And it also meant very few people were on the mean. So most studies look at mean levels. And that's all you care about. If you just look to the mean, very boring. What's really interesting is the movement outside. And this whole idea of personalized medicine is about the individual. In the past, we've ignored it and just squashed them into these means because it's a lot easier for scientists to do that. Make standard deviations easy and graphs, whatever. You don't have to worry about it. But these 10-fold differences had big effects because we also measured some biomarkers of inflammation. This is the immune system getting inflamed. And clearly, when you're getting these glucose peaks or you're getting triglyceride levels that hang around in the blood too long, that some people got and others didn't, that caused inflammation. So we had a clear mechanism that people who were getting regular sugar and fat spikes after eating meals were triggering their immune system, and that meant long-term they're more likely to have inflamed arteries, heart disease, metabolic problems, really immune dysregulation, which explains why it's important, and a predisposition to diabetes. So that, to us, was really important to see, can we predict that and avoid it? And if you did that long-term, you'd avoid these problems that we're routinely facing every day because of our poor diets, and no one had told us about. But the key was that we had all these identical twins because I believed we'd find a strong genetic component, and we didn't. It was about 25% for glucose heritability, and it was under 5% for fat response. So this, by our standards in twin studies, normally everything's over 50%. Even whether you like Mr. Bean or not, gets about 50%. This was an important finding, that even identical twins had different profiles, which meant they had different gut microbes. And people might say, well, why is that? How can they? Well, you've got to remember that the gut microbiome, when you're born, it doesn't exist. You're sterile. So you gain it through the first few years of life. And initially it comes from your mother, it comes from breastfeeding, touching people. If it's cesarean section, the nurse that picks you up, skin microbes. And then you gain a lot of it, in year one were going to the nursery from other kids, or the family pet, or playing in the garden. And these are all quite random. So even identical twins might have been playing in a different part of the garden, or they're playing with different kids in the kindergarten, and they got different microbes. So they ended up really quite different. So even identical twins only share 25% of their microbes as opposed to 20% with you guys. So that's a really important consideration. Then having done that, we looked at the microbiome in detail and worked out that there were links between your microbiome and these responses. So it explained quite a lot of the sugar spikes and the fat spikes based on your microbiome. So we could partly predict what was going on there. And we also thought there was a pattern of good bugs and bad bugs that we thought we could look at across these 1,000 people. And bugs that everybody has. I told you there's lots of bugs you have that no one else has, but you also have quite a few in common. And we've got these 30 bugs, 15 good, 15 bad. They're associated with good health and good diets, and bad health and bad diets. And these are pretty consistent. And so we used that as our new way of scoring things, and also informing about what extra foods might influence these bugs. So that was the very first step into looking at this link between what you eat and the precise microbes that are changing. We've recently updated this, and this was published in Nature a couple of months ago, just before Christmas. And we're nearly 300,000 metagenomes now, so 30 times the number that we had when we did that first study. And again, as I was saying, this is because people in the UK and the US are paying for gut tests and all these other health measures. So we're getting this amazing data set, and 99% of people are signing the consent to say you can use it for research. They're very happy to share it, knowing that the information gets fed back to them and actually improves the advice they get and the personalization they get through the app. What was interesting is that we redid the computation, the database, and we basically doubled the number of species we found in about 4 years. And compared to the previous ones, most of the good bugs we found hadn't been seen before. So when we redid it, just showing how quickly technology is moving, just more precise identification of these microbes, we were able to pick out of the top 50 good ones, most of them didn't even have names. So virtually none of the previous 50 made it. So we're getting much stronger signals, but from bugs that really hadn't looked at before. Basically in medicine, we always look for the bad guys and ignore the good ones. So most of the bad ones were known to us, but the good ones were all pretty unique and hadn't even been given any researcher's name yet. Still waiting for my Spectori microbe. You need to promote it. So we got this ratio and we said, well, how applicable is these 100 bugs across the world? Because we're all different, aren't we? So our sample was from the US and the UK, but we all know that doesn't generalize the rest of the world. But we took all these samples from these countries here, Argentina, Israel, Denmark, France, generally Western countries, but we did have some from Kazakhstan. And in their studies where they'd looked at obese versus non-obese individuals, our ratio worked really well. So we think it's a nice consistent measure. For those of you in the field, you might know all about diversity. Alpha diversity is the way that most people think about gut health. And that's been around for, 25 years, the Shannon Index, maybe longer. And it's basically the richness. It's the number of different species that all of you have. So it worked pretty well. Oh, I've got lots of species, that's good. But it ended up being pretty useless for when you did trials, because it mixed up good and bad ones. You could get, if you're in a trial, you got more good ones, but still your overall numbers were the same because you're just feeding the good ones, you're not feeding some of the other ones. It didn't work very well. So a lot of trials have failed because of our poor methods. Like fiber studies were failing because we were using diversity. But with this new method, it works really well. And we did a number of studies where we had this data showing it's a really sensitive measure of probiotics and prebiotics. And I've got some other examples coming up here. And you can see these longitudinal changes that really reflect, and you can see that those are going up and those are going down. So I think hopefully the field will follow and everyone will be using something like this rather than the old-fashioned diversity one, which I think will really improve the field. And this really helped us in one of our studies because one of the things we believe in at Zoe was that you need this big plant diversity. We produced this packet, which I managed to smuggle through customs. Just between us. Now, I did actually ask them, they said it was all right. Although it does contain something which I'm told is deadly in Australia, a type of mushroom, which apparently is banned by Australia. It's not an edible food, apparently. Every other country says it's fine. But this stuff is a prebiotic. It's 34 different freeze-dried plants. We wanted to, before we launched it, we wanted to actually test it as opposed to just do the marketing. So we did a 6-week intervention, 350 people, and we had 2 control groups, a placebo and a probiotic. It was quite interesting the first time to say, well, how good is a probiotic compared to a prebiotic? Remember, a prebiotic is like the fertilizer for microbes, and a probiotic is like the seeds where you've got live microbes going in there. You're hoping that it's going to have an effect on all those billions of other guys in there. And generally, I think we've perceived probiotics as stronger than prebiotics. May be wrong, but I think that's general perception. We did the study, and we saw big improvements in gut health, mood, energy, and hunger. And interesting, in the first week, those that were in the prebiotic arm got this really big boost in brain effects. So mood and energy and hunger all changed in over 50% of people in the first week. And that was, we were starting to see this in these other studies, which just, to me, really shifted my opinion about the gut-brain axis and really thinking, actually, it's much more important than we've thought. Because generally, in medicine, we don't ask about mood and energy. Doctors hate asking patients about mood and energy because, oh, God, I'd be here all day, talking about it. So we're not taught that that's a reliable thing to do. But with an app recording your mood and energy, as we did this in the COVID pandemic with millions of people, we got pretty good at getting people's responses, and it's very reliable. And so then we realized it is having a really rapid effect on the brain. And it makes sense because most of the nerve, we have this huge nervous supply in the gut. We have this 70% of our immune cells in the gut. What's it there for? It's to tell the brain what's going on. And so if you say, yes, your environment's improving, your diet's improving, it goes straight to the brain and can change your mood. And just to show you the difference between prebiotics and probiotics, we got changes in about 43 of the 100 microbes, either up or down. We got change only about 4 of the probiotic ones. And the controls really were all over the place. So both probiotics and prebiotics worked, but we had about 10-fold bigger effect if you measure it by the number of good or bad microbes that you'd managed to change, which I think is a new way of looking at it. It's much more sensitive than the old way. And if we'd used diversity, we'd have seen no difference. And these are just the symptom changes that I mentioned. We also did a sub-study, a crossover study to show the same effect. And again, much more improvement in symptoms with the prebiotic. Also saw, we did a big we had observational study in 50,000 women going through the menopause. We did a sub-study on 1,000 who were we looked at in much more detail, and essentially the same thing. People are shifting their diet for a gut-friendly diet, going through the menopause, got big reduction in symptoms. The number one symptom that improved most was actually on the brain. So mood and energy was the one that changed by, I think it was 59%. Some really big impact. The others changed, but not as much as that one did, and as rapidly. And we saw that there was an interaction between the gut microbes, diet, and menopausal symptoms. So depending on what microbes some women had, the response to food was rather different, which means that you should be able to personalize this to minimize the impact of menopause. And this just shows there were certain symptoms. So we're hopeful we can get predictions for this and start offering very specific advice for women at the menopause. Like this whole question of eating soy, which is quite a big issue. Soy proteins are metabolized very differently by women depending on what microbes they have. So some might actually cause detrimental effects, but most will have beneficial effects. And depending which microbe setup you've got, you'll get a different effect of ingesting soy. Now, this is our latest paper. We wanted to look at the big data set now. So we're starting to look at over 200,000 people from our data set where we had very good clinical data, microbiome data, diet data. And this was in part also as Zoe wanted to move away from our very gadgety test kit. So if you remember what we based our trial on was a glucose monitor. You had to prick your finger to get a triglyceride level. You had to eat a muffin and do your bloods at 0 and then at 3 hours. And a lot of people didn't like that, as well as collecting your poo sample and recording everything. And so we want to see, can we get the price down? Can we make it much simpler, avoid the blood? And essentially that's what we've done. We've managed to predict the blood sugar levels with the gut microbiome. This is essentially what we've done. So we had really, again, big numbers here. So 3,600 cases. There's twice as many with pre-diabetes. And we had all these people had really big sugar spikes, and we're able to correlate that and predict that with the gut microbiomes. And we had an area under the curve close to 80%, which is pretty respectable as a first go, meaning that we could get rid of the devices and say to people, we're going to predict from your gut microbes, you are sensitive to sugar. You need to do something about it. You need to be not eating the bagels and croissants every day as you have been. Give up the muesli, go on to more high-fat products. And it also, I think, is an interesting one because this could be a very non-invasive test for people going on to develop diabetes. Just the way we do colon testing in stool samples, it'd be very easy to just select a group of these and do it as a population screening of those at risk, because it happens even before you've got a fasting blood sugar level that's high. Vegans, vegetarian, omnivore. Any vegans in the house? One. There's others, perhaps they're too shy. Any vegetarians? Okay. So there's a big anti-vegan campaign in the US at the moment, trying to sort of say that vegans are very unhealthy. We looked at our data set comparing omnivores with vegans and vegetarians, and it turns out that on average, the average vegan has a healthier gut than an average vegetarian, who has a healthier gut than an average omnivore. So well done, vegans. But what was really interesting is if you took the top few percent of each group and you looked at the omnivores, these are meat and dairy eaters, and you took those that ate a lot of plants, they had the best gut microbes. So it's not about whether you eat the meat or not, or the dairy. It's, can you get enough plants on your plate? And I think that's really important. So I think we should be more relaxed about calling ourselves vegans or vegetarians or whatever it is. We should be plantophiles. And realize you can have a very small amount of meat or a small amount of dairy, as long as you're putting other stuff on it. That's probably our optimum diet, because many people believe we are basically omnivores. But in the past, we didn't actually eat much meat, and certainly not what the USDA is telling us we should be eating more of. They recently came out and said we should eat more plants, but basically we're not eating enough beef or dairy. And they already eat twice as much as the rest of the Western world. But the good news is they did say that sugar, artificial sweeteners, and ultra-processed food are bad. So that they got half of it right. So this is just so we found soil microbes in vegans and vegetarians as well. So some of the reasons you're getting that diversity is you're getting these good microbes. No, this is good for you. You should be eating a bit of soil.
You're good.
Again, it's this, but getting out in nature more is probably what we do need. People who don't eat many plants don't get exposed to this, and therefore are going to have less healthy guts. Ultra-processed food. Have you heard of the NOVA classification? A few of you. Okay, it was brought in actually about 15 years ago by some Brazilian epidemiologists to describe food and start to think about food in terms of quality. And they did a very broad brush approach that every country could use. So basically any food with an additive in it or a colorant or anything that's distorted it. And it was very broad. That means that over 50% of the food in Australia eaten, and probably 60% in kids, is ultra-processed using this definition. But it's very broad. Some of this stuff is not very harmful for you because it's just got vitamin C added to it, which makes it an ultra-processed food, but it's not harmful. So there's been a bit of demonizing of all foods when actually we should be a bit more sensible about it. So we wanted to have a graded scale for this, not only looking at additives, but also and focusing on the additives that are known to be harmful rather than any additive, because some additives are healthy, but also look at hyperpalatability, which is the combination of salt, fat, and sugar that the food companies put together to make the bliss point, which triggers dopamine in your brain, in your hypothalamus, to get really excited about food. That's the reason you finish that giant bag of potato chips. This desire to keep eating it. The best highly processed example I know is like the Pringles. One pop you can't stop, or whatever it is. You feel this addictive craving to keep eating. So that's another thing of the feature. And the other is the energy intake rates. They're designed to melt in your mouth, so you don't have to chew them. You can consume huge amounts before you realize what's happened. And these three things are what the companies are doing. And so we wanted to have a score that is an antidote to it, so we can call them out and say what's actually really happening. How do we do this. And so that's gone into our app, and we showed this works, and it correlates with, people who eat these, particularly the worst foods, what we call our category 4. They're the ones that spike inflammation. So we've validated the scale, and it's on our app. So you can go in the UK and the US, and hopefully soon Australia. You can go up and look at any food, and it will tell you not only what's in it, but also what grade of additive it is, which can be helpful because you're looking at breads in a supermarket, you're looking at cheese crackers. It's really hard to work out, and all the front of the label stuff is all bullshit. It's just false labeling, high protein, low calorie, low fat, means nothing, right? But really hard. And then the back of the pack, you need a microscope to see it anyway. So this is why we need help against the food companies. How many people here have at least 3 ferments a day? Okay, about a third of it. That's what you should be eating, really. I didn't really believe in fermentation until I started researching my book on the topic. And up to 3 years ago, the evidence wasn't very good. But there's a really good study from Stanford that showed that inflammation levels dropped by 25% when they were giving volunteers 5 ferments a day. Okay, but that's 25% drop in all your inflammatory proteins is pretty good, damn good. That was compared to fiber. Lots of other studies. So I'm now a big believer. We did a citizen science project with 6,500 Zoe volunteers who weren't into fermentation, asked them to have 3 a day, see how they got on. 1,000 hated it, gave up. Okay, it's real life. Okay, so that's fair enough. They didn't like it, but the others stuck on it. And of those that did, 50% improved their mood, their energy, their hunger. Just like I'm saying, brain first. So it affects inflammation, but that inflammation goes from the gut and tells the brain it's all quiet, we're healthy again. So I think this should be a fundamental that we should all have more fermented foods. They seem to be working on the immune system, not by seeding the gut, which was the old idea that they could somehow take over our gut and that would improve things. You think about it, it's a bit like, you going to a rugby game at the Sydney Stadium and so waving a flag and hoping that everyone's going to notice you. It's like that's if you're like you pretend to be a probiotic, that you get the idea of what it would be like in your large intestine. Anyone here do intermittent fasting, time restricted eating? Yeah, the keen ones on the front row, I see. The same. Intermittent fasting is adapted into time restricted eating, which is not changing your calories, not trying to starve yourself, just eating within time windows. And we did this study. All the studies up to date have been in men, usually young students being paid to do it in American laboratories. Not very generalizable, not many women in it. So we had 150,000 volunteers doing it, mainly women, and see could they hack it for 2 weeks, a 10-hour window, 14-hour fast, get on with it. It turns out a third of them absolutely loved it. They're still doing it. A third of them doing it intermittently. A third of them hating it. And this, I think, is coming back to personalization. We need to be flexible, realize that people are different. Some people wake up really hungry in the morning and want their breakfast. I don't, but I appreciate that other people do. But it's all about listening to your body, listening to that element that's different, rather than saying, like my mum told me, I can't go to school, or work without my egg or my big breakfast. Or, when I was living in Australia, it was often, egg, steak and eggs. Don't need that for many people, but some people might. So to summarize this, we've come up with about 8 principles, which I think are useful. First is to eat mindfully. So just like if you're a vegan or you have strong religious food rules, you don't just mindlessly pick things without working out what's in it. What am I eating? And so you stop, so what am I eating? And then what effects are it going to have on me? May just take a few seconds, but that's a useful thing to do rather than blindly eating in front of the TV or at a buffet or, a cocktail party. Really important. And when I changed my diet, I did go vegan for 6 weeks because it really does help you focus your mind on what am I eating. Eat 30 plants a week. I think it's probably the most important rule. If you do that, you're going to hit your fiber targets without having to count fiber. And that's why we've designed our daily 30 for, difficult weeks when you're traveling or doing something else. But the average Australian prayer is between 10 and 13 plants a week. So it's way off that target. Remember, it's a nut, it's a seed, it's a herb, it's a spice. It's different colored vegetables. It's having fun with it. Reduce processed foods, obviously for the reasons I've said. Don't have to eliminate them totally. Avoid the very worst ones. Don't worry too much about the others. Focus on quality, not calories. So I want everyone to go away and say, the C word is dead. We should just not talk about calories. Vast majority of people, calorie-controlled diets do not work. Counting calories doesn't work. It's a food company trick to get you to eat their foods. Quality is much more important. So whole foods, real food. Reduce your eating window. We've discussed that. Eat the rainbow is, you probably all heard of it. It's all about polyphenols now. We used to call them antioxidants before we knew what they did. Antioxidants is a very fluffy scientific term. It doesn't really mean much. Whereas polyphenols are thousands of these compounds which do have some antioxidant effects, but they also have lots of other effects. But the main advantage for us is they feed the microbes. They're an energy source for your microbes. So brightly colored fruit and veg, berries, lettuces, fruits, and then also the bitter ones, all the bitter flavors, whether it's dark chocolate, olive oil that makes you cough, whether it's red wine that's astringent on your tongue, whether it's dark chocolate, all these are important to get your polyphenols. Pivot your protein. We're not going to get rid of the protein obsession, but maybe we can just modify it so that you get your protein not through big juicy steaks all the time, which are destroying the planet, but you can still have a little bit of your meat if you want it, but you can get so much good protein from beans, pulses, etc., because they also got fiber, they've also got nutrients, they've also got polyphenols, all things that just eating the meat doesn't have. And then eating your 3 ferments a day. And if you do that, you'll be perfect. So summarize, everyone's unique, even identical twins. Our gut microbiome can be manipulated by our diet and our environment. These new gut scores, I think, hopefully will motivate more gut testing and lifestyle change. We really need these big, massive samples. Citizen Science One, and our aim is to get to 1 million pretty quickly. And apps, big data, and citizen science really are the future. And I'll just put an add in here that what Zoe's doing, I think, is trying to lead the gut health revolution. We're an unusual company that's published over 60 science papers in Nature and Nature Medicine, and has a pretty good H index as a company. We've got an app that's free in the US and fairly cheap in the UK. We have a free podcast that gets about 25 million downloads a month, which everyone can listen to, where we get science and nutrition. And we've got gut supplements, and we're going to be bringing other things. So it'd be lovely to get this into Australia if we can sort out the mushroom thing. So if we have anyone who knows anyone influential, we could do that. I'd love to bring it to Australia. And remember, food is the best medicine. And have all these books as well. So the cookbook is a good practical way of doing all these, putting these things into action. But I'll stop there and hopefully get some great questions. Thank you for your time.
Thank you very much. I should say for the people who are online, we don't really have the capacity to take questions via the Zoom portal from there. Tim had indicated earlier that you can send questions in via Zoe if there are things there.
Yeah, I'm not sure I'll be able to answer them, but you can send them in. Hopefully these guys are going to give some great examples.
So Nick will be roaming around. And while Nick's getting the microphone up there for the first question, there is one that might come up from a lot of people. Does beer count as one of your fermented foods today?
It depends the mood I'm in. But some beers, if they've got a sediment in them, and that Guinness did a study that showed it improved cognition. Do you believe it? Yes, definitely takes responsibility. But there is, the thing I didn't mention, I've written a book on fermentation, which I do have a chapter on postbiotics, which is the other thing we didn't mention, which is probiotics that have been killed. And it's a whole new area that just in the last 3 years, they've worked out that dead microbes still have an effect on the body, which has totally changed my views, because I used to say all those pasteurized kombuchas and all that stuff, completely waste of time. But now the science really is absolutely clear that it does work, and we think they work a bit like a vaccine as it goes down into your small intestine, the same way that killed vaccines work. We had them from COVID. And it's just the cell wall and things like this. So potentially cloudy beers with a bit of, dead yeast in the bottom could be healthy for you. So I'm not sure VP or, the counts.
So we'll go, gentlemen.
Foster's definitely doesn't.
How far are you away from the gold standard diet, which everyone says you go to dementia courses, everything else they will say, Mediterranean diet. If you try to define the Mediterranean diet and ask your nutritionist, they all look at you and say, it's a Mediterranean diet, and they won't give it to you. Obesity is inversely proportional to intake of iodine, selenium, or molybdenum. There's studies on all of this. All of these are involved in metabolism of fat, and they're all involved in metabolism of sugar. So when you're doing these studies and everything, you're looking at the intake of those things as being important. The next thing is, if you reckon that the gut microbiome is what you want, why are you not selling gut microbiome?
Okay, a few questions in there. I'll start with the last one. By selling gut microbiomes, I guess you mean poo transplants?
No.
No?
Well, you say there's some really healthy bugs. So your fermentation, you grow the bug, you take your pill and then you see it. I mean, there's a Schaffner reflex in chickens where they walk around the paddock and they sit on a bit of poo and they go, and they suck up the bugs. That's where they get their bugs for their life.
Yeah, I'm not sure that's going to work in Sydney, but
The mindset population.
fecal transplants have generally not lived up to their hype of 10 years ago, and they only really work for 2 or 3 conditions. One, they work for non-responders for immunotherapy and cancer, interestingly, so helping the immune system, which is a life and death issue, actually, and they're routinely doing this in the US. And I think if I was on, stage 4 melanoma and immunotherapy, I'd want the very best microbes to make me respond to it. The other is ulcerative colitis, works in about 1 in 5 people to cause remission. So, but it seems to be quite limited, probably because we've all, a bit like, we haven't worked out who the perfect donor is, and we're all very different. I suspect it's that. It's been much harder. And we used to think it could cure obesity with getting good microbes. That hasn't worked at all. The other question about, are we looking at selenium and these elements and nutrients in foods? The answer is no. We've moved away from that reductionist view, because you can chase thousands of nutrients and come up with an observational story about them. Our view is that if you take a more holistic view, like eating the way we're describing, over 30 different plants, fermented foods, mainly plants, whole foods, no processed foods, you will naturally get all these nutrients in that diet without worrying about it. If you obsess about one, you're going to miss out on another. And I think my view is the nutrition world has been obsessed with the reductionism because that's where the commercial sales have been. And we need to move away that a much holistic view. I agree, though, we haven't really defined the Mediterranean diet at all.
But the trouble is that I need to leave all of
we should move on because otherwise
you don't activate B2, you don't conversation. K, D, B12, E. So if you don't have those, you can't activate. So you can't deal with glucose.
Okay.
So the lady in the green, and can I please ask the people keep your questions really short so that there's an opportunity to move around with others.
Yeah, thank you so much. And thank you, Tim. That was such an amazing talk. I just wanted to ask, at the beginning, you were talking about the nutrient pack that you brought with you and snuck through customs, which was really interesting to see. And that'd be great if that was in Australia. And you did some research around that versus a control. But then later in the presentation, we're talking about really good, healthy eaters, like the omnivores who had the lots of salads. And so they were in a good percentage. And then afterwards, you gave us some tips in order to really increase our nutrition. So if you were how does your research, I guess, deal with really good, healthy eaters, people who are following your advice otherwise, people who are probably on those scores, eating lots of healthy salads, in terms of if they need the nutrient or how they compare against people who are taking your supplement pack?
Okay, well, it's always harder to study people that are, way above average healthy to see how you can optimize their health. It's much easier to see the changes in people who are suboptimal when you start. They have the bigger changes, they have the most microbiome changes. It's just basic statistics, really. It's if already we score people out of 1,000 for our gut test. So if you're 750, it's hard to shift you significantly higher than it is for someone who's starting at 250. So that's where we need 1 million people to really start getting enough people in that top group to say, how do we personalize things to them who are already doing a pretty good job? What we do have is links between certain microbes. So we've got these 50 good microbes, and we're now slowly getting to know which foods trigger those microbes to grow. So in the future, we will be able to give you more precise instructions. Say, well, I'm supposed to be eating more watercress. I find it hard to get watercress, but I should be eating it because I'm lacking some good microbes that we know are associated with watercress. So these are things that will happen. But it's just harder to deal with the people who are already very healthy. But hopefully, trial and error and people retesting themselves, they can do this themselves.
Yeah.
So, Erin, shout out.
I don't need a microphone.
Oh, but other people won't be able to hear.
I just have a question about your fermentation study. So you said it was people that consumed at least 3 ferments per day. What was your control for that study? Because
there was no placebo control. So it wasn't a randomized controlled trial. This is 6,500 people. It's an open study. So all we had was the people that dropped out and didn't do it and report it. So it's not at the same level as a randomized controlled trial. I was doing it more as a pragmatic study. There are lots of small randomized controlled trials showing it works. This was to say, well, how feasible is it to tell people to get on and eat 3 ferments a day, and what effect does it have in the broad run? It's not gold standard, but I think we need both of those kind of studies. 20 people studied intensively and then thousands done in a population.
we'll take this lady here next. How much more time do you have?
I'm okay. I can run a bit over if we have time.
Thanks so much for your talk. I just want to ask you about your sample. It's an awesomely sized sample. But from what you said, it's the people involved are all paying for it. So therefore they come from a quite high socioeconomic area of the population. I wonder if you could say something about how you feel that affects the results, the study. How do you compensate for that and so on?
Well, we're not trying to compensate because we're not claiming it is generalizable to the whole population. We're saying it's generalizable to people who have enough money to buy a test. As the numbers get bigger and bigger, we still get people who have terrible gut microbes, people with different diseases, people without colons, people with, and so this enables us to build up this database. It's the same with genetic studies. If you looked at 23andMe, again, you had to pay, you have $100 to spend on health. So you were different. But despite that, we're seeing a huge range in gut health. Mm-hmm. And we also see differences between countries. The UK is pretty bad, we think, but it's much better than the US. So the same age person, they're about 30% lower on average than us. So I think we can still learn a lot from it, even if we're not getting the very worst people yet. But some people, will offer it to their, 18-year-old son who's on a terrible diet, or a student. They're pretty poor diets. So I think if we get the numbers big enough, we'll be fine.
If you pass the microphone, then while you're passing, just one you can give a yes or no to. Is the microbiome test actually an approved diagnostic anywhere as yet?
No.
But
the reason is if you go to try and get medical certification, you can't change that test for years, and it takes about 2 years of paperwork. So all the cutting edge technology in the medical area is in the wellness space, because we can keep upgrading our algorithm every week to improve it. Any medical test is locked in time, and we need to change the rules because it stops us doing that when it could be actually very useful. We have a test for cancer, for example, we're not allowed to use because we'd be labeled as a medical device. We'd have to shut down everything we do. It's really counterintuitive about where this is going.
Thank you so much, the organizer. I flew 6,500 km here from Kuala Lumpur to Sydney to ask this question. Professor Tim, Australia actually have the toughest biosecurity, the custom officer. So I'm just wondering, for this trip, what did you bring in your ferment to hit your 3 ferments a day?
Turn off the recording for a second.
Well, I declared everything because my last stop was in Tokyo, and I went to a fermenting museum where they had. So I have some natto in my bag, and because it was live, it didn't need cold storage, and it's in a sealed bag. So I've got natto. For those who don't know it, fermented soybeans, which is one of the highest concentrations of microbes. It's got koji, which is a fungus, plus bacteria in it, and that should get me through and until I can. I've only got a week in Australia to survive, so I'll be.
After we've got an afternoon.
He was quite happy that the customs officer was very happy with natto for some reason.
I'm acutely aware, because I can see all the hands go up, there's a lot of people who are bursting to ask more questions, but we do have some afternoon tea refreshments out there. If you can cope with being swamped face to face, that might be timely to move out and take that opportunity for a more personal questions there. So I apologise for those who didn't get to ask their question here now, but thank you all for coming. I'd like to remind those as part of our normal attendance that the Nutrition Seminar Series is an ongoing thing that's approximately monthly. Our next one is in March, I believe, with Margaret Orman Farinelli as the speaker. So hopefully we get similar sized audience for that, maybe not as many online. But once again, thank you very much, Tim, for